right, welcome to the deep dive. Today we are cracking open a stack of sources you've shared with us, focusing on a genus of bacteria that holds some really surprising significance, especially as you navigate your studies and look towards your future practice. We are diving deep into the world of Bacillus. Now, let's be honest, for you, a dental student, Bacillus might not be the first name that springs to mind when you're thinking about microbes lurking in the mouth or, the usual suspects causing cavities or gum disease.
Right. Probably not top of the list for oral infections specifically.
But as these sources make abundantly clear, understanding Bacillus is actually fundamental, particularly for mastering a vital, kind of non-negotiable area of your foundational knowledge in clinical practice.
That's precisely right. While the bacteria responsible for, say, caries or periodontal issues are certainly crucial for you to focus on, the sources you've provided really highlight how a genus like Bacillus, which seems incredibly widespread and perhaps a bit abstract initially.
Yeah, just out there, right?
Yeah, exactly. It actually plays this critical, almost behind-the-scenes role in something as vital as infection control. Understanding its unique properties isn't just academic. It's absolutely essential for patient safety in any clinical setting you'll be in.
Absolutely essential. So our mission today, guided by the material you've shared, is to unpack these sources, pull out the most important insights about Bacillus, what it looks like, what some species can do, how labs find it, the little bit the sources tell us about how it's treated, and crucially, why it matters specifically in a clinical setting like dentistry. Okay, let's unpack this. Let's start with the basics. What are we looking at generally when we talk about the Bacillus genus according to these sources?
Okay, so the sources introduce Bacillus as a really large and diverse genus containing nearly 50 different species, but they immediately highlight the defining characteristics that kind of group them together. First, they are gram-positive.
Okay, the gram stain, key differentiator.
Absolutely. Tells us about their cell wall structure,
Right.
They are also rod-shaped, which is, where the name bacilli comes from, it simply means rod-shaped bacteria.
Simple enough.
But perhaps the single most important feature the sources emphasize, and it's a theme we'll keep coming back to, is that they are typically sporing. They have this amazing ability to form endospores.
The spores. That's like their superpower for survival, right?
Yeah.
That sounds really important.
It really is. And the sources tell us just how incredibly widespread this sporing ability makes them. They're described as being widely distributed in nature.
Meaning
Found commonly, almost universally, in environments like soil, dust, air, and water. They are literally everywhere around us.
Wow.
Their ubiquitous nature is a defining feature of the genus as a whole. And the sources give us another clue about their prevalence and hardiness when they mention that Bacillus species are often isolated as contaminants in bacteriological culture media.
So they just show up uninvited in the lab sometimes.
Pretty much. If they can easily float in on dust or air and start growing on your lab plates, even when you weren't trying to culture them, it really underscores just how tough and environmentally persistent they are. They're, masters of survival outside a host.
That point about contamination is really practical for thinking about laboratory work. You always have to be mindful of unintended microbial guests popping up.
Definitely.
Now, the sources then seem to zero in on a specific species that is particularly important and well described within the material you've shared, Bacillus anthracis.
Right, B. anthracis.
It's singled out not just for its clinical importance, but also its sheer size compared to many other bacteria. The sources call it one of the largest pathogenic bacteria ever described.
Yeah, it certainly stands out morphologically. The sources provide specific dimensions, giving us a pretty concrete picture of its size. 3 to 8 micrometers long by 1 to 1.3 micrometers wide.
So 3 to 8 long, 1 to 1.3 wide. How does that compare?
Well, to put that into perspective, consider that many common bacteria, like E. coli or staph, are significantly smaller, maybe less than a micrometer in width, and just a few long. So B. anthracis is quite robust in size for a bacterium.
That's a chunky microbe. And beyond its size, the sources highlight its shape and appearance as quite distinctive when you look at it under a microscope.
Definitely. It's gram-positive, as you'd expect for the genus, and non-acid-fast. It's described as a straight, sporing bacillus, often appearing quite rectangular in shape.
It's rectangular. Interesting.
That rectangular appearance is part of its characteristic look, especially when grown in the lab. And here's where it gets really interesting. The sources give us this vivid description of how B. anthracis looks when you grow it in culture and examine a stained smear. They don't just appear as individual rods.
No.
No, they arrange end to end in these long chains, and the ends where one bacillus meets the next aren't smooth or rounded. The sources use the terms truncated or swollen concave ends to describe these junctions.
Truncated or swollen concave ends.
Okay. And when you see these long chains of rectangular rods joined by these distinctive ends, the sources say it creates that memorable, almost visual shorthand. The bamboo stick appearance.
Oh, the bamboo stick thing. I've heard of that description.
Yeah, exactly. This morphology is a classic identifier if you're viewing a smear from a suspect culture.
Bamboo stick is a great way to visualize it. Those segmented chains must look really unique.
Mm.
Now, another key point the sources make, one that helps differentiate B. anthracis from many of its bacillus relatives, relates to movement, right? Motility.
Yes, exactly. The sources explicitly state that B. anthracis is non-motile. It doesn't have flagella or other mechanisms for active movement.
So it just sits there, unlike most other bacillus.
Pretty much. That lack of motility is a simple but effective test you can perform in the lab using a motility medium. Seeing that it doesn't spread out from the inoculation line helps distinguish it from other bacillus species that are motile, which is, the majority of the genus.
Okay, good distinction. Let's circle back to those crucial spores, because their characteristics are also important for identification, aren't they?
Absolutely vital. The sources emphasize the details of the spores themselves. They are described as oval, or ellipsoidal, a specific non-spherical shape. They are refractile, meaning they appear bright and shiny when viewed under a light microscope because light bends around them, indicating their dense, tough nature.
Okay, shiny ovals. Where are they in the cell?
They are located centrally within the bacterial cell, and critically, the sources state they do not cause the mother cell to swell.
Ah, so they fit neatly inside without bulging the cell wall out.
Exactly. This non-swelling feature is another important diagnostic clue. Some bacteria form spores that bulge out the cell wall, making the cell look sort of like a drumstick, but B. anthracis spores form neatly inside without distorting that rectangular rod shape.
Okay, central oval non-swelling spore. Got it.
This combination is quite characteristic. The sources also specify where these tough spores form, which is a vital detail about the bacterium's life cycle and how infection spreads. They state spores form in culture, in soil, and in the exudates or tissues of dead animals.
Dead animals, okay. Not living ones.
Right. But, and this is a really important clinical point, the sources state they never form within the blood or tissues of living animals. The form actively replicating and causing disease inside a living host is the vegetative non-sporing rod. The spores are the environmentally stable survival and transmission form.
That's a key distinction. The spore is the tough traveler. The rod is the one doing the damage inside a host. Makes sense.
Mm-hmm.
And what about that other protective layer mentioned? The sources talk about B. anthracis forming a polypeptide capsule.
Yes, it forms a polypeptide capsule, which is actually somewhat unusual. Most bacterial capsules are polysaccharides, sugars.
Oh, right.
This capsule is specifically made of D-glutamic acid. The sources state this capsule surrounds the entire chain of bacilli when they are in animal bodies or under specific culture conditions.
Like what conditions?
Like if you add bicarbonate to the culture medium or grow it under 1025 per CO2, which kind of mimics the conditions inside a host body.
A capsule made of an unusual amino acid.
Yeah.
That sounds like a pretty sophisticated defense mechanism for the bacterium when it's trying to infect something.
It absolutely is a major virulence factor. That D-glutamic acid capsule is poorly recognized by the host immune system, particularly the phagocytic cells like macrophages that usually engulf bacteria.
So it helps it hide.
It makes it much harder for these immune cells to grab onto and destroy the bacteria, allowing B. anthracis to multiply rapidly and cause widespread infections, like an invisibility cloak almost.
Wow. Okay, so let's quickly recap the morphology based on these sources. We're talking about large, gram-positive, rod-shaped bacteria, often forming those characteristic bamboo stick chains with truncated ends. They are spore-forming, and these spores are oval, central, don't swell the cell, making them incredibly hardy and persistent in the environment.
Right, super tough.
And B. anthracis, specifically, is non-motile and forms that unique polypeptide capsule made of D-glutamic acid, which is key for its ability to cause disease in a host.
Exactly. This foundational understanding of their structure and survival mechanisms, the spores, the capsule is absolutely crucial before we discuss what they do do and how we encounter them. It sets the stage.
Right. It's like understanding the armor and the survival kit of the microbe before you talk about the battle. Okay, so let's pivot from what they look like to what they can actually do. What does the source material tell us about the pathogenesis, the ability of these Bacillus species to cause disease in humans?
Well, the sources are quite focused here, narrowing down this vast genus to highlight the species most associated with significant human illness. They state that 2 species of Bacillus are primarily associated with significant human morbidity.
And those are?
First, Bacillus anthracis, which we've just spent time describing morphologically, and second, Bacillus cereus.
Okay, let's start with B. anthracis. The sources say it's the causative agent of anthrax. And it's not just one type of disease, right? The sources specify it can manifest in different forms depending on how the spores enter the body.
That's right. It depends on the route of entry. You have cutaneous anthrax affecting the skin.
From handling stuff?
Yeah, often from handling contaminated animal products like wool or hides. Then there's gastrointestinal anthrax from eating contaminated meat, which is rarer but more severe. And inhalational anthrax from breathing in spores, which is the most severe form and historically the one associated with bioterrorism concerns.
And it can even spread further.
Yes, the sources mention it can even lead to anthrax meningitis, an infection that spreads to the membranes around the brain and spinal cord.
Oh.
So that's a pretty wide range of potential diseases from just one organism.
Yeah, that covers a lot of ground. Serious stuff.
It can affect multiple body systems. And the sources also highlight B. anthracis' significant place in the history of microbiology, which is quite interesting.
Oh, so.
They mention it was the first pathogenic bacterium to be observed microscopically. Imagine Robert Koch or others seeing these rods in infected animal tissue and linking them to the disease for the first time.
Wow, that must have been groundbreaking.
Absolutely. And building on that, it was the first communicable disease proven to be transmitted via inoculation of infected blood. Koch's postulates were largely developed using anthrax as the model. It was key evidence that helped solidify the germ theory of disease. So these historical points really underscore its importance in understanding how infectious diseases work.
That's fascinating historical context. It's not just some random bug. It's a historical figure in germ theory development.
You could say that, yeah.
And getting back to the structure, the sources explicitly state here in the pathogenesis section that its ability to form that unique polypeptide capsule contributes to its pathogenicity.
Right, they confirm it.
It confirms that the armor we discussed earlier isn't just for show. It actively helps the bug cause disease.
Exactly. That capsule is a crucial factor in its virulence, allowing it to evade the immune system, establish infection, and spread within the host. It's a primary weapon.
Okay. Now let's turn to the other major player the sources mention in terms of human disease, Bacillus cereus. This one sounds a bit more familiar, maybe, often linked to food safety issues.
Right, be serious. Yes, this one is probably more familiar to most people. It's frequently implicated in food poisoning.
I know it's notorious for issues with foods like cooked rice that have been left sitting out at room temperature, right?
Yeah.
Allowing spores to germinate and the bacteria to produce toxins.
That's the classic scenario, the fried rice syndrome. But the sources indicate it's capable of causing problems far beyond just a transient bout of food poisoning, which can actually have 2 forms. One causing diarrhea, one causing vomiting, depending on the toxin.
Oh, okay. 2 types of food poisoning even.
Yep. But beyond that, the sources state it's recognized as an important pathogen not only in these gastrointestinal infections, but also in serious ocular infections.
Eye infections. From Bacillus.
Yes, eye infections, which can sometimes be quite severe and even lead to vision loss, especially if introduced via trauma or contaminated contact lenses, for example. And even more severe, systemic conditions like catheter-related septicemia.
Bloodstream infections from IV lines and stuff.
Exactly. It's a bloodstream infection often introduced via medical devices. And also endocarditis, which is a dangerous infection of the heart valves.
Wow, so B. cereus is surprisingly versatile and potentially dangerous, not just a food bug.
That versatility and capacity for severe non-GI disease is something the sources highlight, underscoring that B. cereus isn't just a minor food contaminant. Especially in healthcare settings, it can pose a significant threat as an opportunistic pathogen, gaining access to the bloodstream or sterile sites through catheters, wounds, things like that.
Okay, good to know. The sources also briefly mention a couple of other Bacillus species.
Yes, just briefly. They mention Bacillus licheniformis as being associated with some food poisoning cases as well, so another potential foodborne issue within this family.
Okay, and Bacillus subtilis. What about that one?
Yes, Bacillus subtilis. The sources describe this one as generally less pathogenic than B. anthracis or B. cereus. However, and this is an important concept for you as a future clinician, it may act as an opportunistic pathogen.
Opportunistic, meaning it takes advantage of a situation.
Exactly. It typically doesn't cause disease in healthy individuals with intact immune systems and barriers, but it can seize an opportunity when the host is vulnerable, maybe immunocompromised or has a wound or a medical device in place. The sources specifically link Bacillus to causing eye infections and septicemia in these opportunistic scenarios.
That distinction between primary pathogens, like B. anthracis maybe, which can cause disease in healthy people, and opportunistic ones, like B. subtilis, which need a helping hand, is really key in understanding microbial threats, isn't it?
It's fundamental. It means that even generally harmless bacteria found in the environment can become dangerous in the right circumstances, especially in a hospital or clinical setting.
Yeah.
And the sources add that other Bacillus species, beyond the ones explicitly named, can also be involved in various opportunistic infections. This just reinforces the idea that while we focus on the most notorious species, the potential for problems exists across the genus, especially in vulnerable patient populations.
Okay, so let's sum up the pathogenesis section from these sources. The genus Bacillus contains species capable of significant human disease. You've got B. anthracis, causing the severe multi-system disease anthrax via different routes, and B. cereus being a common cause of food poisoning, but also capable of serious eye and bloodstream infections. Other species, like B. ligoniformis, can cause food poisoning, and B. subtilis and others can cause opportunistic infections in vulnerable individuals.
That covers the key players mentioned here.
Now, importantly for our listener, while these sources don't specifically call out Bacillus as typical residents of the oral flora or common culprits behind cavities or gum disease.
No, they don't list them in that context here.
Right. But understanding the general principles of bacterial pathogenicity outlined here, how bacteria cause disease, the different ways infections spread and manifest, from skin lesions to systemic sepsis, the role of virulence factors like the capsule, and that crucial concept of opportunistic infection is absolutely foundational for you as a dental student.
Absolutely. It provides the essential framework, the mindset for recognizing, preventing, and ultimately managing any bacterial infection you might encounter, even those caused by organisms you haven't specifically studied in detail yet.
It builds your microbial intuition almost. Gives you that broader understanding of the potential landscape of microbial threats you need to be aware of in a healthcare setting. Okay, so if you suspect a Bacillus infection, maybe anthrax or maybe B. cereus food poisoning, or perhaps you're investigating a potential source of contamination in the lab or environment, laboratory diagnosis is essential to confirm its presence and identity. The sources provide a clear outline of the steps involved in finding them in the lab.
Yes, they describe the typical microbiological workflow quite well. It starts, naturally, with obtaining the right sample specimen collection.
And what kind of samples are we talking about?
Well, the sources give examples depending on the suspected infection. For general culture, if you suspect cutaneous anthrax, for example, clinical exudates like pus or fluid from a lesion are collected. And specifically for B. cereus food poisoning, the diagnosis relies on isolating the organism from relevant specimens.
Like the food itself.
Exactly. Any remaining suspect food that was eaten, or if available, patient vomit or stool samples. This really highlights that the sample type is crucial and has to be directed by the clinical presentation. You look where the problem is.
Makes sense. You need to look for the organism where it's causing trouble or where it entered the body. After collection, the sources move to preparing the sample for viewing under the microscope.
Right. Microscopy is a primary step. Smears of the collected specimens are made on glass slides and then stained using various methods. The sources list Graham's method, which, as we mentioned, is fundamental for bacterial classification and initial identification.
Gram-positive rods, right?
That's what you'd expect to see. They also mention McFadden's method and Gemsa stain. These different stains help visualize the bacteria and sometimes specific structures, like capsules, which might not show up well with a simple Gram stain.
And what do you typically see with that Gram stain from a clinical sample if Bacillus is involved, based on the morphology we discussed?
The sources confirm it typically reveals large Gram-positive bacilli. Seeing those large, purple, rod-shaped bacteria would immediately suggest the possibility of a Bacillus species and warrant further investigation.
Okay, and if you suspect the presence of a capsule, particularly relevant for B. anthracis, in a clinical sample from, say, blood or tissue, how do you see that?
Right. The capsule itself doesn't stain well with standard stains, but the sources describe how it can be visualized as a clear halo or zone surrounding the bacterium by using India ink staining.
India ink, like for drawing.
Kind of, yeah. It's a negative staining technique. The India ink particles stain the background darkly, making the unstained capsule visible as a bright, clear ring around the bacterial cell. It's quite effective.
Cool. Is there anything more specific?
Yes, for more specific and sensitive confirmation, especially for B. anthracis, the sources mention a direct fluorescent antibody, DFA test. This test uses antibodies tagged with fluorescent dyes that specifically bind to the B. anthracis capsule itself or detect its polysaccharide cell wall antigen. When you look under a fluorescent microscope, the bacteria light up brightly if the specific antigen is present.
So that's a much more definitive identification, even from a direct clinical sample.
It's a very powerful tool for rapid specific identification, yes.
Okay, so combining different staining techniques and specific antibody tests allows for both general identification as a bacillus and more specific confirmation of a particular species like B. anthracis. But microscopy alone isn't usually enough to pinpoint the exact species or get enough material for further testing, like antibiotic susceptibility. That's where culturing comes in, right? Growing them.
Precisely. Culture is the next essential step. Clinical specimens are inoculated onto various types of laboratory media, the nutrient gels or broths designed to support bacterial growth and help in identification.
What kinds of media do the sources mention?
They list common general media, like blood agar and nutrient agar, which allow a wide range of bacteria to grow. But they also mention specialized media, specifically PLET medium and nutrient broth.
PLT medium. What's special about that?
The sources highlight that Knisley's PLT medium is used for the selective isolation of B. anthracis.
Selective isolation. What does that mean in practice? How does it work?
It means the medium contains specific ingredients, usually antibiotics or other chemicals that encourage the growth of B. anthracis, while inhibiting or suppressing the growth of many other bacteria, especially other spore-forming bacteria, like other Bacillus species that are commonly found in environmental samples and might contaminate a clinical specimen.
So it gives B. anthracis a competitive advantage on the plate, making it easier to find if it's present, especially if it's mixed with lots of other bugs.
Exactly. This is particularly useful when trying to isolate B. anthracis from samples like soil, environmental swabs, or even some clinical samples where you expect a mix of bacteria. It ensures that you can detect low numbers of the target organism without it being completely overgrown by everything else.
That makes sense. And how long do you typically need to wait for these bacteria to grow enough to be visible on the plates?
The sources state that plaques are typically incubated at 37 degrees C, which is standard human body temperature and optimal for many pathogens for about 18 hours.
So usually an overnight incubation.
Yeah, an overnight incubation usually does the trick for many relatively fast-growing bacteria like Bacillus.
Okay. After incubation, you look at what has grown on the plates, and the sources emphasize that the appearance of the colonies, the visible clumps of bacteria, is a key diagnostic feature, especially for B. anthracis.
Absolutely crucial. For B. anthracis, the sources describe colonies with a very characteristic morphology, specifically calling them Medusa head colonies.
Medusa head.
Huh.
We talked about the bamboo stick for the individual bacteria. Now Medusa head for the colony. That's another really evocative description. Can you describe what that actually looks like on an agar plate?
Yeah, it's quite distinctive. Imagine tangled, sort of grayish-white, unruly hair. The colonies of B. anthracis often have these irregular, sort of wavy, filamentous outgrowths extending from the edge of the main colony. It gives the margin a rough, almost swirling or C-shaped appearance that resembles, well, a Medusa's head of snakes or tangled locks of hair. It's quite different from the smooth, perfectly round edges you see with many other bacterial colonies.
Okay, I can picture that. Very irregular edges. What else is important about the colonies, especially on blood agar?
Another crucial characteristic for B. anthracis, especially when grown on blood agar, is that the colonies are non-hemolytic.
Non-hemolytic. So remind us, that means they don't cause lysis or breakdown of the red blood cells in the blood agar.
Exactly. Blood agar contains sheep or horse red blood cells, and many bacteria produce enzymes or toxins called hemolysins that break down these cells, creating a clear or greenish zone around the colony. The sources specifically state that B. anthracis colonies lack this hemolytic activity, so there is no clearing or discoloration around the colony on blood agar. It just grows right on top without affecting the red cells underneath.
And why is that important?
Because this absence of hemolysis is a key differentiating feature between B. anthracis and most other common Bacillus species, many of which are strongly hemolytic, causing large zones of clearing. So seeing a large gram-positive rod growing in a medusa head colony that doesn't like B blood cells is highly suggestive of B. anthracis. It's a critical negative result that helps confirm the suspected identity.
That's a powerful negative test, actually. Looking for what isn't happening is just as important as looking for what is.
What about growth in liquid media, like the nutrient broth mentioned? Does that show anything special?
Yes, in nutrient broth, the sources note that B. anthracis typically shows two features, a pellicle formation and a deposit. A pellicle is a thick, sort of cohesive film or scum of bacterial growth that forms on the very surface of the liquid medium, indicating aerobic growth right at the air-liquid interface. And a deposit simply means that some bacterial cells also settle and accumulate as sediment at the bottom of the tube. Okay, so a surface film and some stuff at the bottom. Got it. So you've got these macroscopic clues from the colonies on agar and the growth in broth. What's the final step in identification, according to these sources, after you've cultured it?
After you have these characteristic colonies or growth patterns, the sources state you would prepare another gram-stained smear, this time taking a sample directly from the growth that you've cultured on the agar plate or from the broth.
And what are you looking for there?
Looking at these cultured bacteria under the microscope, you would expect to see those characteristic tangled chains of large gram-positive bacilli that we discussed in the morphology section. The bamboo stick appearance might be even more evident in culture. And importantly, especially if the culture conditions allowed for sporulation, which might happen on nutrient agar as nutrients deplete, the sources note that some of these bacilli will contain those central oval, non-bulging spores, confirming their presence and characteristic appearance.
Okay, so laboratory diagnosis, based on these sources, isn't just one single test. It's really a multi-step process that builds evidence piece by piece.
It's exactly.
You start with the right sample, you examine it microscopically with various stains, maybe including special stains for capsules or those specific fluorescent antibodies. Then you grow it on different media, including selective ones like PLE if needed. You look very closely at the appearance of the colonies on the agar plates, checking for medusa head, checking for hemolysis on blood agar, and then you confirm with a final microscopic look at the cultured bacteria, checking for those telltale chains and spores.
Precisely. The sources explicitly state that the definitive identification of Bacillus species, particularly B. anthracis, relies on combining all these observations. These specific colonial characteristics like the medusa head appearance and lack of hemolysis on blood agar, along with the detailed microscopic view from the stain smears of both the original clinical specimen, if possible, and the cultured growth. It's a comprehensive classical microbiological approach, putting all the puzzle pieces together.
This multifaceted approach to laboratory diagnosis, starting from sample collection through different levels of microscopy, culturing on various media, including selective ones, and observing both macroscopic and microscopic features of the cultured organisms, is really fundamental diagnostic microbiology. It's the same kind of logic and set of techniques used to identify a wide range of bacterial pathogens, whether you're looking for Bacillus or, Streptococcus or Staphylococcus or something else entirely.
Absolutely.
So this section provides a vital framework for you, the listener, for understanding how you might confirm the presence of a microbial threat, or how you'll rely on laboratory services in your future practice. It's how the detective work gets done.
It's building that practical understanding of how microbes are detected and identified, moving beyond just recognizing symptoms in a patient. It connects the clinic to the lab.
All right, so we've covered what they look like, what some of them can do, and how labs find them. Let's touch briefly on how they're dealt with treatment. The sources seem pretty brief on this part, is that right?
Yes, that's correct. The information provided in the specific sources regarding antimicrobial treatment is quite limited. They don't go into much detail here.
Okay. What do they say about treatment then? It seems the only specific mention of a treatment approach is for one particular scenario involving Bacillus.
Right. For gastroenteritis caused by Bacillus cereus, the food poisoning aspect, the sources state that treatment is primarily symptomatic.
Symptomatic treatment.
Yeah.
Meaning you just treat the symptoms.
Essentially, yes. It means the focus is on managing the symptoms the patient is experiencing rather than necessarily administering specific antibiotics to kill the bacteria. For B, serious food poisoning, which is often caused by preformed toxins in the food
Mm,
or toxins produced quickly in the gut rather than a deep tissue invasion.
right.
The main issues are typically vomiting and flare diarrhea, which can lead to dehydration. So, symptomatic treatment involves supportive care, like rehydration with fluids and electrolytes, maybe anti-nausea medication if needed.
Mm.
The illness is often self-limiting.
Meaning it just goes away on its own?
Yeah, it usually resolves on its own within a day or so as the body clears the toxins or the bacterial load decreases naturally. Because of this, and because toxins are often the main driver, antibiotics are generally considered unnecessary or ineffective for the acute symptoms of typical B-serious food poisoning.
Okay, that makes sense for the food poisoning. But what about the more serious infections we discussed, like anthrax caused by Bacillus anthracis, or those severe B serious eye or blood infections? The sources are silent on specific drug treatments for those.
That's right. Based on the text provided, the sources do not detail specific antibiotic names, dosages, or regimens for treating anthrax or other types of invasive infections caused by Bacillus anthracis or other Bacillus species. While treatment protocols definitely exist for those serious conditions, they aren't outlined in this particular set of materials.
So, based only on the material you've shared with us today, we cannot go into the specifics of antibiotic protocols for these more severe Bacillus infections. The text simply doesn't contain that level of detail on treatment.
Exactly. It appears the primary focus of these sources, in terms of depth, lies more in describing the organism itself, its morphology, its pathogenesis, and critically, how it's identified in the lab, which actually leads us directly to the most important connection to dentistry presented in this material.
Ah, okay. Which brings us to the most critical section for you, the listener, our dental student. We've explored the general microbiology of Bacillus. Now let's connect it directly to your field, dentistry. Section 5 is all about the implications of Bacillus in dentistry, according to these sources. This feels like where it really hits home for you.
Absolutely. This is where all that foundational knowledge about morphology, particularly the incredible resilience of the spores, becomes immediately relevant and intensely practical for day-to-day dental practice. The sources are very clear and direct about this primary connection.
What do they say?
They state, the most direct implication of Bacillus species in dentistry relates to infection control and sterilization procedures.
Infection control. Okay, that is, without question, absolutely paramount in any healthcare setting, and especially dentistry, where procedures often involve exposure to blood, saliva, aerosols, lots of potential for transmitting pathogens.
Couldn't agree more. It's non-negotiable.
So how exactly does Bacillus fit into this vital area based on what the sources tell us? Why Bacillus?
This is the critical link that makes understanding Bacillus essential for you, even if it doesn't typically cause cavities. The sources state that spores of a specific Bacillus species, Bacillus stereothermophilus, are specifically utilized as biological indicators.
Biological indicators, like a test.
Exactly. They're used to test the effectiveness and efficiency of sterilization processes, particularly those carried out by autoclaves, which use steam under pressure. This is the core connection highlighted in these sources.
The spore test. This is something you will definitely encounter and perform routinely in practice, right? The sources refer to this as biological monitoring.
Yes, biological monitoring is the technical term. It means using live, highly resistant biological entities, in this case, bacterial spores, to challenge and verify that the sterilization process actually worked and killed everything. And the sources are quite specific about the recommended frequency for this monitoring.
How often?
They state that these biological monitors, or spore tests as they are commonly called, are recommended for weekly use to verify that sterilization is being achieved reliably in a dental practice.
Weekly use. Wow. That recommendation from the sources underscores just how vital this validation step is. It's not just a one-time check when you buy the machine. It's an ongoing routine procedure to ensure that the equipment you rely on every day to make instruments safe for patients is consistently working correctly.
Precisely. It's quality assurance for patient safety.
So how does this process work? Based on what the sources describe, you're placing something containing these Bacillus stereothermophilus spores inside the sterilizer chamber.
Correct. You place these standardized biological monitors, which typically consist of a small strip of paper or a self-contained vial, containing a known high population of dried B. Stereothermophilus spores inside the autoclave chamber, along with a normal load of instruments or materials you are sterilizing.
And where do you put them? Just anywhere?
No, the sources offer specific guidance on where to place these monitors within the chamber for the best test. They suggest placing them particularly in areas where achieving sterility is most challenging.
Okay, that makes perfect sense when you think about how an autoclave works. With heat and steam needing to get everywhere, you want to test the hardest to reach spots, right? Like the coldest part of the chamber, or maybe deep inside a wrapped cassette.
Exactly. You want to test the areas that are slowest to heat up or slowest for the saturated steam to penetrate. Because if the sterilizer can successfully kill the highly resistant spores in those tough spots, you can be confident it's killing everything, even less resistant microbes, throughout the entire load everywhere in the chamber.
So you're essentially challenging the autoclave in the most rigorous way possible, using these super tough spores in the hardest place to sterilize.
That's the idea. You're testing its ability to reach and maintain the lethal conditions, the right temperature for the right amount of time, with saturated steam, even in the least favorable locations within the chamber. Then, the sources imply that after the sterilization cycle is complete, you would retrieve these spore monitors and process them.
To see if the spores survived.
Right. To determine if any of the B. Stereothermophilus spores survived the cycle. While the sources themselves don't detail the process of culturing the spores from the test strip after the cycle, which typically involves placing the strip in growth medium or activating a self-contained vial and incubating it to see if the spores germinate and grow,
Okay.
they do strongly emphasize the critical importance of the results of this test. A failed test, meaning the spores grew, indicates a potentially serious failure in the sterilization process that needs immediate investigation and correction.
And what makes Bacillus stereothermophilus spores the absolute ideal test organism for autoclaves specifically? Why not some other tough spore, or maybe B. anthracis spores?
That's a great question, and it ties directly back to that initial discussion about morphology and the nature of spores and the specific sterilization method. Autoclaves operate using moist heat, high temperatures, typically 121 degrees C or even 132 degrees C, under pressure using saturated steam to kill microorganisms.
Right. Steam sterilization.
Now, bacterial spores, as we discussed, are the most resistant forms of microbial life known. They have incredibly tough coats and dehydrated interiors that make them resistant to heat, chemicals, radiation, and drying, far beyond what vegetative bacteria, fungi, or viruses can withstand.
The ultimate survivalists.
Exactly. Now, among the spore-forming bacteria, Bacillus stereothermophilus, which, by the way, is now technically named Bacillus stereothermophilus, though the sources use the older, more familiar name, so we'll stick with that.
Okay, stereothermophilus.
It's particularly known for producing spores that exhibit extremely high resistance, specifically to moist heat. They are much harder to kill with steam than, say, B. anthracis spores, or spores from anaerobic bacteria like Clostridium.
Ah, so they're specifically super resistant to the exact killing method used in an autoclave.
Precisely. Therefore, if an autoclave cycle is successful in killing B. stereothermophilus spores, which are among the most challenging microbes known to kill with moist heat, you can be highly confident that the cycle has also killed all other potentially pathogenic microorganisms that might be present on your instruments. That includes less resistant spores, all vegetative bacteria like staph, strep, mycobacteria like TB, fungi, and viruses like hepatitis B, C, or HIV. They are the benchmark.
So their incredible resilience to the specific method used by autoclaves of moist heat is exactly why they are the universally accepted standard benchmark for verifying that the sterilization process is effective. It's a perfect direct application of their fundamental biological survival mechanism.
Right.
It's not just any tough bug, it's the toughest bug for that specific process.
It is a crystal clear example of how understanding basic microbial characteristics, like spore formation and specific resistance profiles, has direct, critical implications for ensuring patient safety through routine procedures like sterilization monitoring. The sources also emphasize that the results of these biological monitoring tests should be meticulously recorded.
Documentation. Always key in healthcare.
Absolutely. It provides proof of proper functioning and a record for tracking issues. And the sources highlight specific times when the results of these spore tests are particularly crucial and must be verified and documented.
When would that be?
First, when commissioning new autoclaves, you need to run tests to ensure the brand new unit meets performance standards right out of the box before you ever use it on patient instruments.
Makes sense. What else?
Second, after any servicing or repairs, if someone has worked on the machine, you absolutely need to confirm that it's functioning correctly again and still achieving sterilization temperatures and times before putting it back into service.
Okay, new machine, after repairs,
Oh,
any other times.
yes. Thirdly, as part of training for new staff, ensuring that everyone who operates the autoclave understands the entire sterilization process, why validation with spore tests is so important, and how to properly perform the monitoring, interpret the results, and document everything is critical.
This really drives home that sterilization in dentistry isn't just a passive process of pushing a button. It's an active, validated procedure that requires routine verification using these incredibly tough bacterial spores and careful documentation every step of the way. It's a core component of patient safety and quality assurance in any practice.
It really is, and it's the extreme resistance of Bacillus stereothermophilus spores that provides the necessary reliability and confidence for this essential validation process.
Now, just to be crystal clear for our listener, based on the material provided, this application in sterilization monitoring is the main, perhaps only, direct connection of the Bacillus genus to your future dental practice discussed in these sources, right?
That's what the sources indicate, yes. They explicitly state that the text do not indicate that Bacillus species are common oral commensals or direct causative agents of typical oral infections within the provided text.
That's an important clarification, especially for you as a dental student, since your focus is naturally going to be on oral microbiology, the bugs causing cavities, gum disease, abscesses, etc. You're not typically expected to be diagnosing or treating infections caused by Bacillus within the mouth itself, like caries or periodontal disease, based on these specific sources.
Correct. While the sources mention their potential as opportunistic pathogens systemically or in other sites like the eye, their primary and clearly stated relevance in a dental context derived solely from this text is unequivocally their essential role in ensuring the efficacy of sterilization protocols using B. stereothermophilus spores.
So this single application, while maybe less clinically dramatic than treating a severe infection like anthrax, is actually foundational to patient safety and effective infection control in every single dental practice every single day.
Absolutely foundational. And understanding why these particular spores are used connecting back to their incredible ability to survive the harsh conditions of moist heat, which we learned about when discussing their morphology and resistance, is key to appreciating the scientific basis and the critical importance of this routine procedure you will be responsible for.
It really does bring it full circle, doesn't it? From the fundamental microscopic structure and survival strategy of these bacteria, those incredibly tough spores, to a practical, essential, potentially life-saving procedure you will perform or oversee weekly throughout your entire career. It shows how seemingly abstract microbiology concepts have direct, tangible impacts on clinical practice and patient well-being.
That's a perfect way to summarize the connection.
So, wrapping up this deep dive into the Bacillus genus based on the sources you provided, we've seen that this is an incredibly widespread and hardy group of bacteria, defined by their rod shape, being gram-positive, and most notably, forming those incredibly resilient spores that allow them to survive really challenging conditions out in the environment.
Mm-hmm. We've touched on a few species causing significant human disease, particularly B. anthracis, responsible for the serious disease anthrax via multiple potential routes of infection, and B. cereus, a well-known cause of food poisoning, but also capable of more severe systemic infections like ocular infections and septicemia. We noted that other species, including B. subtilis, can act as opportunistic pathogens, causing issues in vulnerable individuals or when they get into normally sterile sites.
We walked through the basic steps of identifying them in a laboratory, starting with collecting the right clinical or environmental sample, looking at it under the microscope, using different stains like grams, India ink for capsules, or specific fluorescent antibodies, then culturing it on various media, maybe selective PLET medium,
Right.
and observing those distinct macroscopic features like the medusa head colonies or the critical lack of hemolysis for B. anthracis, finally confirming with microscopy of the cultured bacteria, looking for those telltale chains and spores.
And we highlighted the most direct and absolutely critical link to dentistry presented in these sources: the essential role of Bacillus stereothermophilus spores as the gold standard biological indicator for validating that autoclaves are effectively sterilizing instruments. This underscores the non-negotiable importance of effective infection control in preventing the transmission of all potential pathogens in the dental setting, even those not specifically mentioned as typical oral flora in this text.
Yeah, it reinforces that sterilization isn't just about setting a timer on a machine. It's a scientifically validated process that requires regular verification using the toughest possible biological challenge available for that method.
Exactly.
This deep dive, drawing solely from the material you provided, really demonstrates how understanding even seemingly distant microbes like Bacillus contributes to a broader, essential knowledge base required for safe and effective clinical practice. It's particularly relevant in foundational areas like preventing infection transmission through rigorously validated sterilization protocols. It shows how it's all interconnected.
It really does. And maybe this raises an important question for you to think about as you continue your studies. Considering how incredibly tough Bacillus spores are, and the fact that their extreme resilience is precisely what makes them the universal standard for challenging and verifying our sterilization equipment, what does that imply about the baseline level of microbial resistance we need to account for and actively overcome in any clinical environment to ensure true patient safety? It makes you think about the challenges posed by even tougher potential threats, or prions perhaps, and the constant need for vigilance in infection control, even beyond what these specific sources detail about Bacillus pathogenesis itself. Something for you to perhaps ponder.